All right, so today we'll be talking to you about brain structure and function in boys with teens with Klinefelter syndrome. My name is Laura Foland-Ross. I'm a neuroscientist and senior research scholar at Stanford, and I work with Alan Reiss on the study of brains, genes, and puberty, or the BGAP study. Today, we're really excited to show you some of our initial findings. So I'm going to break today's talk down into 4 broad sections. First, I'm going to speak a bit about Klinefelter syndrome, what it is, and common behavioral features. I'll then launch into our initial findings of brain structure and function in KS. I'll then talk a little bit about possible influences of incomplete pubertal development on the brain and associated behavior in KS. And then we'll have Alan Reiss wrap up with conclusions and take-home findings. Okay, so on to what is KS. So as many of you likely know, KS is a genetic condition that's associated with an extra X chromosome. And it's the most common sex chromosome condition, occurring in as many as 1 in 448 males. And KS is associated with a variety of physical features, including extra breast tissue, tall stature, and infertility later in life. But it's also associated with incomplete pubertal development. That begins around age 13 or 14, although it can vary widely between individuals. In addition to physical features, there's a variety of different behavioral features associated with KS. These are experienced by many males with KS, but not all, and this range in severity of these features can also widely vary between individuals. So these can include motor dysfunction and impaired visual motor integration, learning-based learning issues, as well as alterations in a domain of cognition known as executive function. And this includes a variety of different cognitive functions, such as working memory, so memorizing a phone number in your mind, sustained attention, which involves a sustained focus on this talk, for example. And inhibition is another component of executive function, and this corresponds with the ability to suppress an impulse to do or say something, for example. And all of these different behavioral features can really show up in the classroom, with boys and teens with KS potentially having some difficulties with academics. And so being able to get a clear picture of the types of behaviors that boys and teens experience living with KS is important, because it allows us to intervene sooner and improve outcomes. So this is a multi-site study. So here at Stanford, we have our team directed by Dr. Alan Reiss. And on the East Coast, we have our co-principal investigator, Dr. Judith Ross, whose labs are at the Nemours Children's Hospital in Delaware and Thomas Jefferson University. And this research is funded by the NICHD. So far, we have acquired data from a total of 73 boys and teens with KS and 69 typically developing age-matched controls. We are in year 5 of the study, so we are just wrapping up. All of the participants in our study are between the ages of 8 and 17. And at each of multiple time points, we do in-depth assessments of puberty, behavior, and the brain. So with the brain, we look at both brain structure and function. And we do this using magnetic resonance imaging. You can see one of our research participants here is just finished with his mock scan. So you can see him sitting in our mock scanner, which is what we use to get all of our participants very comfortable with the sounds and the feels of the actual MRI scan. Okay, so now I'm going to jump into our findings of brain structure and function. These are findings from cross-sectional analyses that we've conducted of boys that have completed data collection at baseline assessment. So here's findings from our structural analysis. So these are areas of the brain that show differences between boys and teens with KS and typically developing boys. And these are areas of the brain that are known as gray matter, which is the part of our brain that contains neurons and synapses. And you can think of this as a fingerprint of the effects of KS. And so what I want to draw your attention to are the areas that are in blue. These are areas of the brain that show reduced gray matter volume in males with KS compared to typically developing males. And these areas, which include the insula, the inferior frontal cortex, the striatum, and the amygdala and hippocampus, among other regions such as the temporal lobe, are all areas that are involved in behaviors that we see affected in KS. So these are things like learning and memory, emotion regulation, behavioral control, and language difficulties. So these findings, which we are submitting for publication, are going to undergo additional analysis with our behavioral measures so that we can better understand the direct correspondence between alterations in the brain in KS and their effects on corresponding behaviors. We also looked at brain function. One way that we looked at brain function is how the brain is metabolically active at rest. So here we simply have our research participant lie awake in the scanner, and there's no cognitive demand. We just have them rest comfortably with their eyes open. And what this allows us to do is understand which areas of the brain co-activate or activate together at the same time and in the same way. And this gives us information about how different brain areas are functionally connected to one another. And what we see is that in our KS cohort, there are widespread reductions in the connectivity between brain regions. And so looking at this further in association with behavior, we see that the degree of connectivity differences is significantly associated with behavior. So boys that have greater reductions in functional connectivity between brain areas also show the most pronounced alterations in executive function, social behavior, and symptoms of anxiety and depression. So this suggests that there is a direct association between what we're seeing in the brain and behavior. We also looked at brain activation in a different way using a cognitive challenge. And so for this type of investigation, we have our participants look at stimuli that are projected onto a screen. So there's a little mirror placed over their head here, and they can see everything that we can project onto a computer screen. And we also give them a button box so that they can respond using button presses to different pictures or stimuli that we show them. And here's what we showed them. So this is called the go, no go task. And it's a task where the subject simply sees a letter projected on the screen. And the only instruction in this task is to press a button on their button box every time they see a letter, except for when they see a letter X. So when they see a letter X, don't press at all. And this sounds very simple, but it's very challenging because the letters appear very rapidly. And we really load them up with lots of letters that aren't the letter X. So this gets them into the habit of forming an automatic or prepotent response where they are very used to pressing, and they really have to show a lot of cognitive control to withhold that press when they see that X. Another thing to know about this task is that it's pretty long. So this lasts 18 minutes. And so boys and teens in our study really have to exert a lot of attention and focus to be able to stay engaged in this task. And what we see in our neuroimaging results is that boys with KS show reduced activation in specific areas of the brain that are involved in sustained attention and impulse control. So this includes areas such as the right inferior frontal gyrus here and here, as well as the insula and the caudate. So all of these regions together are really important for executive function. And this suggests that there may be a neural basis for the alterations in executive function that we see in boys and teens with KS. Okay, so now I'm going to just leap over to some interesting findings that we have on potential influences of incomplete pubertal development on the brain. So specific areas of the brain are very densely populated with receptors for testosterone. And these are areas that are really important for executive function, including the prefrontal cortex, but also the hippocampus and the amygdala. The amygdala is more so involved in emotion, but together these areas are very important for emotion regulation and impulse control. These areas, when exposed to testosterone at puberty, bind that testosterone at the receptor, and this triggers dynamic waves of neural remodeling in adolescence. And what we see in our study is that boys and teens with KS are very similar to their typically developing counterparts in early ages. So when we look at total testosterone, we see that the two groups are not very different at earlier ages, but at around age 13, boys with KS appear to show a reduction relative to the typically developing group in the amount of testosterone that they produce. And this is also seen in measures of testicular volume, which is a more stable measure of pubertal development. So we see at earlier ages, while the two groups are not very different, at older ages, beginning at around 12 or 13, the two groups really begin to diverge. And this is caused by a slower growth rate of the testes in males with KS. And what we find in incorporating these measures of puberty is that boys with KS that have a lower testicular volume for age, so this is testicular volume for their age, comparing testicular volume to population norms for typically developing males. What we see is that boys with KS that have lower testes volume also have the highest scores on this measure of attention difficulties. And here, higher scores indicate greater difficulties in attention. So this suggests that incomplete pubertal development, the more severe patterns of incomplete pubertal development you have, the greater difficulties in attention you also experience. We also see these patterns when we look at activation in the brain. So boys with lower testicular volume and lower testosterone also show lower activation in these areas that we target with the go-no-go task. And so that wraps up my presentation of initial findings from the BGAP study. I'll now turn it over to Alan Reiss, who will wrap up with some conclusions.
Thank you so much, Dr. Foland-Ross. It was a great presentation, and I just want to point out to everyone who's viewing how much Lara has meant to this study. It really could not have happened, in particular the brain part of it, without her very energetic, thorough mastery of the area. So thanks so much for that. And again, to thank Dr. Judith Ross, who's my partner over on the East Coast. Judy and I have worked together for, it's amazing to say, a decade. So we couldn't have done this study without her and without the two groups working together. So I'll just wrap up in the last couple of minutes and just point out something that's really obvious, I'm sure, to everybody, that all learning and behavior arises from the brain. And so we wanted to be sure that we completely assessed as much as possible with our current technology, brain development and function in KS. And that brain development and function in KS that we are showing as different from typically developing boys is likely to be associated with both genetic, in other words, X chromosome gene expression factors, as well as hormonal or testosterone factors. So we have to always keep that in mind with a condition such as Klinefelter syndrome. Further, though, it gets even more complex. These genetic and hormonal factors interact with other genetic factors, not necessarily related to the X chromosome or Klinefelter syndrome. And of course, the environment. We never should forget the impact of the environment on outcome, because it's really quite massive. So environment, when I refer to it, I think about prenatal environment, school, goodness of fit of the school, home environment, family function, peer environment, acceptance, rejection, neglect, etc. All of these factors together combine and interact to affect outcome. So what do we do? Given this knowledge that we are generating in the study, in addition to what we already know about KS? Well, we should aspire, both in families and professionals, to optimize those factors, those individual factors that we can influence, that we can change. Testosterone, school environment, home, opportunities for successful interaction with peers, study habits, etc. But also work to establish a better understanding of those critical relationships among genes, brain development and function, environment and behavioral outcome in persons with KS. And we have taken our best shot at it in the BGAP study, and we hope to continue to generate information and papers and presentations that will be useful for the families as well as people who are professionals working with boys and families with KS. So what can we hope for in the future as we gain this knowledge? Well, there is a concept called precision medicine, and precision medicine means that you have a particular approach for a particular area of a condition or a particular diagnosis. And I think we should be hopeful that we can bring precision medicine approaches for persons with KS in the future. So how do we do that? Well, first we have to understand KS-associated variations in brain development and function, and as I've been saying already, how those are associated with genetics, hormonal environment, and other aspects of the environment. That approach, I believe, is our best hope for improving outcome and quality of life. And I'm not just thinking about pie in the sky here. I work with other families and other research projects where precision medicine approaches are already available for a limited number of genetic variations or conditions, such as sickle cell anemia. Now, of course, sickle cell anemia it does involve the brain, but not as its primary impact. But already stem cell transplants are available for people with sickle cell anemia that have the potential, and I've been seeing this, to actually cure the condition. Okay, sickle cell anemia is more tractable, kind of easier to understand, and doesn't really is not as complex as KS. But the hope there is that a similar type of approach, not with stem cell transplants, but understanding all those different aspects that can give us a pathway for precision medicine. There's a very, very recent drug approved for Rett syndrome, which is, can be a very devastating condition, primarily affecting girls. And a drug was just approved, trifenotide, that is meant to preserve and develop abnormalities of cellular and synaptic development in this condition, and hopefully reduce symptoms and improve development. So this is the type of treatment that I hope is going to be available on the horizon for boys with KS, along with more precise environmental, school-based, et cetera, treatments. And with that, I'm going to stop and thank everyone who participated in this study. The families have been marvelous. Come to see us during the pandemic or seeing us by video. And if you are interested in participating in the future, please take a snapshot of the slide and put your information in. We were hoping that we'd get to do this research for another 5 years. We put in a grant to continue the study, so cross fingers for us. Thank you very much.